First, the impulse reaches the synaptic bulb. Then the calcium enters the bulb. The vesicles then migrate to the cell membrane. The vesicles then fuse with the cell membrane. Neurotransmitter acetylcholine is released into the synaptic cleft. Acetylcholine diffuses across the cleft and then binds with the receptor proteins. The motorized plate depolarizes and action potential moves along the sarcoplasm. The impulse travels to the neuromuscular junction, colored green. Then the action potential moves along the sarcolemma, down the transverse tubules, and into the sarcoplasmic reticulum. And calcium is then released from the sarcoplasmic reticulum. This is the sarcomere, with the Z line in blue, the actin in silver, and the myosin in gold. The calcium that is released into the sarcoplasmic reticulum binds to the end of the tropomin. Tropomyosin moves and exposes the actin binding site. The myosin head extends and binds with the actin. The ADP and phosphate are released from the myosin head, causing the myosin to contract and pull the actin filament. The ATP comes in and binds with the myosin head, causing the linkage to break, and the ATP is broken down into ADP and a phosphate. This is repeated many times by numerous myosin heads on the actin. This will cause the whole sarcomere to shorten, also known as muscle contraction. For relaxation, acetylcholinesterase breaks down the acetylcholine. This loss of actin potential causes the calcium ions to be transported back to the sarcoplasmic reticulum. While the myosin head is bonded to the ATP, it is relaxed and the cross bridge is reset. The lack of calcium ions allow the tropomin and tropomyosin to block the actin binding sites, inhibiting any contraction.